Thanks for those talks there. It's really a pleasure to be here, to have this opportunity. My name's Luke Noon, and as Tom said, I'm a researcher here at the CIPF Biomedical Research Institute in Valencia, Spain. And my interests are liver regeneration and the impact of metabolic disease on the liver and its ability to repair itself. I'm also the co-founder of a new mixed 3D electron microscopy unit that we established here just last year. And this integrates complementary infrastructures and expertise from two very different research institutes. So our own, which is a basic science institute here in Valencia, where our EM facility has expertise in array tomography. And we've sort of forged this partnership with the fantastic imaging facilities led by Jose Gomez at the Isabial Hospital. So we're kind of joined forces and sort of making, it's not changing the screen here. We're making sort of things stronger together, and we're looking for collaborations with people interested in array design, imaging, and volume electron microscopy. So I'm going to talk about volume EM workflows in liver. The title is Thinking Outside the Brain Box. This is Valencia, where we're situated. And this is a statue that I cycle past every day on the way to work. And I found that out the name of the statue is Points of View. And I think it's great because it kind of highlights how volume EM kind of changes your perspective on things. Certainly, I see these axons sort of popping out of everywhere. And I really love what this technique is doing for our research. Okay, so our objectives in the lab is to really try to pioneer a sort of new concept, which is peripheral nervous system connectomics. So we've heard two fantastic talks about connectomics in the brain, and you can see the power from those talks of the connectomics approaches and how far we've got really in tissues like the retina and in the brain as well, and the olfactory bulb, as we just heard. And these advances have gone hand in hand with advances in volume electron microscopy. And what we'd like to do is to apply these to the peripheral nervous system. And you might ask why. Obviously, the peripheral nervous system is nowhere near as complex as some of the circuitry that you've seen in the previous talks. But somatic nerves aren't these, they're not just these sort of simple, faithful connections from, let's say, a somatic nerve to a muscle cell. We have autonomic nerves, somatosensory nerves, and they have these large arbalized arrays, their nerve termini, and they can have quite interesting patterns of connectivity with different cell types. And also, there are whole organ systems in the periphery which we know close to nothing about. So the liver is one such organ, and we have only a very rudimentary understanding of where hepatic nerves actually terminate in the liver. And we know that if we cut the nerves to the liver, we actually prevent the liver from regenerating properly, which was our particular interest. And also it affects glucose homeostasis and liver function. So what we're trying to do is to apply volume EM and connectomics to systematically map the hepatic nerve network and also generate workflows that can be applied elsewhere in the peripheral nervous system. To give you an example of what we're doing, here's an axon in the axon I showed you earlier. And if I play this video, you can see we can sort of click through the segmented axon and see where it makes contacts with different cells. Here at the top, it's making contact with a cholangiocyte in a bile duct. And then lower down, it's actually coupled to a hepatocyte. So two different epithelial types interconnected by the same axonal fiber. And as we zoom out, you begin to get a bit of an idea of the kind of complexity of the nerve network around the portal vein. So if you see here, you can see most of the nerves are around the afferent vasculature. So here is the portal vein, which brings blood from the gut. You have hepatocytes here, a bile duct, and you have all of these axonal fibers in amongst these connective tissue that we're trying to begin to understand. So where are we? Well, we've actually pretty far advanced with this. We've databased thousands of individual contacts. We have these interactive datasets, which enables you to click and link back to each contact. And this enables us to build up kind of tissue level profiles of the relative connectivity of the peripheral nerves in the liver under healthy conditions, and then when the liver is injured or during development, etc. We're also tracing axons, as I've mentioned, and this gives us an understanding of the kind of pairwise contacts with different cell types along the length of axons in this kind of en passant way. And these connectograms, we're seeing a shifting as the liver is injured and responding to injury, suggesting remodeling of the hepatic nerve network. And finally, we're also reconstructing large areas of these volumes, looking at the kind of hepatic neural niche. And this is giving us an insight into the kind of symmetry within the network and clues as to how different tissues may be integrated together by similar nerve endings. Okay, so a little bit different to what you've seen so far in the central nervous system, in the brain. You have to struggle to not find nerves in the brain, right? It's kind of like a spaghetti soup of axons. You can see the challenges of actually trying to untangle that. Our problem in the peripheral nervous system is slightly different. You can actually, if you just randomly apply volume electron microscopy to different places in the liver, you'll unlikely find an axon or a nerve. So a targeted approach is essential to locate these sort of sparse, localized networks of nerve fibers and nerve termini within our volumes. And so we've been working very hard to refine correlative volume electron microscopy workflows. In this case, we're using a transgenic mouse model that guides us to the glia around the axons in the liver. And to give you an idea of the workflow that we've developed so far, well, like many array tomography workflows, they're kind of modular. And obviously, array tomography has its advantages and disadvantages. And one of the things I really like is this kind of modular workflow that you can have, because it enables us to integrate expertise from our collaborators in different institutions. And that gives us a scalability. And it means that as a small lab, we don't have to be experts in everything. So to give you an idea of how this workflow has evolved, well, and how it works, we have this first step, which is fixing and finding. So you section the liver, we scan it by confocal microscopy to get an idea where the nerves are. Then we have a very clear pattern, a very clear readout of where all the nerves are in our final included EM sample. And this is something that we've developed. Actually at UCL, I went to the Lloyd lab. They are fantastic investigators in peripheral nerves, and Jemima Burden and I work very closely together to get this going. The next step is making your tissue arrays. And this is something that we've refined here at the Príncipe Felipe in Valencia. And Mario is a fantastic electron microscopist who we've been working with here. And we're generating really high quality tissue arrays of 300 to 400 serosections. We can do that routinely. And so we have our tissue arrays. We put them on a single beam scanning electron microscope. And the next step is to pick the regions of interest from within each slice prior to acquiring. So this is actually an important step to highlight. It's not just a question of sticking it on the microscope and acquiring everything. We have to pick these particular regions of interest, and that we use our information from our correlative approaches to do that. Having acquired those regions of interest, the last step is to align, and that gives us our kind of aligned volume. And then the final step is which is not trivial, is to do the axon tracing and start the segmentation and to really get an idea of the nerve network within each region of interest that's been acquired. And to give you an idea here, it's slightly a bit of a sort of the comparison with the brain. Well, maybe the liver is not the most intelligent organ. Within this sample, we have 4 millimeters of axons that have been skeletonized. That equates to about 125 meters of axon per millimeter cube. So a lot less than in the olfactory bulb, I imagine, but you know, there's nerve there to be studied. And our data sets go into our WebNosos account and where we can work with them. And I'll just give you an idea of what this data set looks like, if it will let me open it up. Here we have our axons have been skeletonized. We can move around very nicely and easily in it. We can zoom in and out, and then we can trace we've traced all our axons through with these skeletons. We also have the contacts added. And so that's what our data look like when we have them in our database. Okay, so the next step is what we do with each axon. So we annotate the axon using skeletons, and we also label nodes that are branch points and axons that actually, or axon branches that terminate within the volume. And we use separate skeletons to annotate the contacts with different cells within the liver. And the links function within WebNosis is very convenient for us copying and pasting these reference points into these spreadsheets. So we generate these really nice interactive spreadsheets where each axon has, we can go back and find all of the features where they make contact with different cell types. And then each region of interest would also be summarized in terms of certain statistics about branching, the total length of axons, the density of nerve termini, and also tissue descriptors. So it's important to know that wherever you sample, you might get different amounts of tissue, and these kind of descriptors give us a good idea of how the network relates to the tissue around it. So I'm moving on to the workflow and the data challenges that we've faced in setting this up. I've mentioned the technical challenges of peripheral nervous system connectomics, and really we're right up to speed with, we can make it's quite easily, let's say for 6 animals, we can make arrays in a month or 2. So we've got this bit really up and running. I did mention that we started this in London with Jemima, and then I moved the lab. I set up a lab here in Valencia, as Tom mentioned. That created an immediate problem, which is that we didn't have any on-site access to an SEM. But this was in 2022, and right at that point, Jemima had started a pilot with WebNosos. So this was very convenient, and she started putting our data sets into WebNosos, and I carried on annotating. I started annotating and analyzing them in this way. And then as we refined the tissue arrays that we were making and that approach, we literally just put them in the post and send them to Jemima. And she would put them on the scope. So this very quickly became clear that it was a good way to work between different institutions, very effective. And so we've kind of over the subsequent years, we've expanded this kind of model. I should say that we can switch between institutions quite easily. So Jemima's account and mine, we can switch it within WebNosos to gain access. And we've kind of grown this. So recently we've connected with another great group of people in Sweden. Julia Fernandez runs a fantastic facility there with Rafa, and we've started working on a project that looks at nerves during development in the liver with Emma Anderson. And as I mentioned at the beginning, we've also last year set up this mixed unit, which actually gives us remote access to their microscope. But I should say that really we don't need that, because despite the fact we've got access to 3 different facilities and we have this remote access, it's really important the expertise of the people with the microscope. So we haven't really got fully up to speed with driving the microscope ourselves. And actually we've started to use WebNosos regularly for our workflow integration at this point. So if we send an array to Rafa, he'll put it on the scope and he'll acquire some low resolution data sets, put them into our WebNosos account. We'll annotate quickly and guide him to where we want to set up our image acquisition. So that's a really useful point about what we use our WebNosos account for. To give you an idea, obviously the workflow has evolved a lot since we started. And this workflow evolution has led to a data revolution for us. So we're small, and a small lab, big data problems, because it's kind of got more and more as we've gone along. The project actually started, we were using serial section TEM, Jemima and I, at the very beginning, and then she switched and we switched to array tomography SEM. And you can see our progress in terms of the volume capture with these methods has been exponential. So this is an exponential scale. We've got a nice exponential increase with time. This gives you an idea of some of our better serial section TEM samples, and you can see you've got a few axons in there, and we've really sort of boosted this, and we're really working at a different level now. And the more nerve, the more structure, the more volume, the more nerve, the more structure, and the more targets. So that's the volume that we're acquiring. But I also wanted to make the point that we've kind of plateaued as regards the size of the data set. And you might ask why. Well, why have we sort of we're acquiring bigger volumes but at slightly lower resolutions. So in order that we can sort of save not data set size, but actually time on the microscope. And that's a point that I thought was important to make. And Rafa has been fantastic in helping us to do this kind of optimized acquisition. And that's because imaging is still a bit of a bottleneck. Even though we've optimized the setup with our sort of our workflow, and we've reduced the beam time here by playing with the acquisition settings, it's still, a single array may take several days to acquire and may be occupying the scope for several weeks. So that's something to consider when you're working with maybe people who have other users and other demands. It's occupancy on the microscope is quite an important thing for to take into account, even if we ignore the costs, which are kind of shocking when you start to think about those sorts of things. And obviously, if you want to carry on with this exponential increase, we're going to have to make the shift to a kind of multi-beam approach at some point, if anyone's interested in working with us with that. Okay, so the bottleneck imaging, but we've done our best to overcome it. Another bottleneck is alignment, and early on that is a problem. Was more of a problem. Now it's less of a problem. Obviously, array tomography data requires more work with alignments, and you get more artifacts, as you probably know. This is one of our earlier data sets. It had lots of problems, not very well aligned. And one of the recent things that we've started to do is use the AI-based alignment tool in WebNosos. And we actually went back to this old data set, which we hadn't annotated because it wasn't good enough. And we realigned it by skeletonizing features of interest throughout the volume to give some fiducials for how to align. And then we went through it, and it's really shown to be very effective in kind of recovering data sets that were previously not up to scratch. So alignment, this is something we're kind of incorporating into our general workflow. We normally get the data sets off the Atlas software after the basic alignment, and then we will use this alignment program to do it. Finally, the main bottleneck, I'm slightly jealous of some of these AI-based tools that you guys are using in the brain. Obviously, the liver and the array tomography, the tissue is different. It's not quite so straightforward to automate everything at this stage, given that we're the only people working on the liver, I think. But annotation is laborious, and it's essentially all manual still. So these axon skeletonization steps, the feature segmentation, if we need it, the databasing, manual, and then the connectomic analysis, it's all kind of manual. Switching to WebNosos has helped us in a major way, in the sense that each person in the team can work together on the data sets, and they can use their own laptops, or you can work from home. So that really helps. You don't have to go to a workstation to do these sorts of things. So this has been a revolution for us. But you can see the hours that have gone into our data. We've got thousands of hours, hundreds of working days, and in click time on WebNosos, we have over 1,000 hours of clicking and segmenting time. So that's a lot of work, but it makes our data sets really rich for future streamlining and training of AI tools. So at the top here, to give you an idea of when we started, we would be manually segmenting axons that we thought were interesting enough to segment, because they'd all been skeletonized. And then we've recently, since it was introduced, the AI tool for segmentation, where you literally just click on, or you can draw a box around, that will segment the axon for the next sort of 16 slices or 20 slices, depending on how you play with the settings. So this has really been a step forward, and is beginning to improve what we're doing. But where we want to move to is automated segmentation based on our axon skeletons, and using our data sets to train AI. And we're hoping that we'll be able to get tools that will recognize where these varicosities are, which will give us an idea of where the contacts are within our axon skeletons. And then also something we're working on developing or hoping to develop soon is automated target cell recognition. So to get an idea of the different cell types that are around those varicosities within our volumes, that should all help to speed our workflow. So to sum up, we've developed this nice modular workflow that's enabled us to pilot this peripheral nervous system connectomics. And I haven't talked about applicability to other tissues, but essentially because we use Schwann cells, we can apply this to any tissue really that has peripheral nerves in it. And we can go to those nerves and begin to do these sorts of experiments. And this is in the liver is giving us new insight into the remodeling of the nerves in response to injury and in disease states. I hope I've been able to illustrate that collaboration is the key to scalability. Our collaboration with webNosos has been an example of that. And I've also pointed out the main constraints that we've faced and we're still facing, this manual annotation, and also the volume capture with single beam availability being an issue. Our integration of webNosos has helped in many ways, but it's certainly enabled us to do this kind of remote and collaborative sharing of data, makes the data very interactive. We can optimize the setup of the microscope using this approach as well. We have team-based annotations with lots of people working on the data sets, efficient sharing and databasing of features. The databasing is something that I really like, that you can copy these links and go back to exactly where you want to go in the volume. And then we've also begun to integrate the kind of new AI-based tools that webNosos have brought online recently. Finally, our future directions. Well, I think we're at an exciting point where AI and connectomics can sort of redefine how we understand peripheral nerves. And we certainly hope to be able to leverage our annotated volume archive that we put all of this work into to kind of be able to train AI to automate some of these things for the future. And whilst at the same time acquiring bigger and bigger data sets, I think that's a sort of common thread that you want to have more and more. And hopefully we can capture this full nerve network around the portal vein, because we don't have to go any further. It's kind of restricted, but we're nowhere near being able to get a full data set. And finally, applying this to human samples is also something we're working on. So thanks for your attention, and I'm happy to answer any questions that you have. I should, of course, acknowledge all of the people that have worked on this. Alison and Jemima, fantastic, and without them, this wouldn't have got started. Mario, who's done all the work with the arrays, and our collaborators, Julia, Emma, Jose, Isabel, and Deborah, and all of the team who've been annotating, and of course, Norman, who's been very responsive, and we've worked very well on the things that we've done to now. So thank you, Norman.